Hi, my name is James Hickman. I'm the co-founder with Mike Schuler of Hesperos. I'm also the chief scientist, and I also have a dual role as I'm a professor of nanoscience technology, chemistry, material science and engineering, biomolecular sciences, and electrical engineering at the University of Central Florida here in Orlando. Today I'm going to talk to you about human-to-chip systems for use in therapeutic index determination and systemic toxicity in preclinical drug discovery applications. So just a really quick overview for Hesperos. It's a company founded again by Mike Schuler and I. Mike Schuler is considered the founder of the field of body on a chip. We've licensed a number of patents from Cornell, where he's a professor, and UCF, where I'm a professor. Mike and I won the Lush Prize for Return of the Animals. We use a lot of SBIR funding to keep Hesperos going. Now we work with a lot of national, international pharma companies. We have a 14,000 square foot facility down here in Orlando. Got 50 people at the company. We're a CRO. We figure these systems are too complex to be able to send them out and have people learn how to use them in 2 weeks. More importantly, we are the first microphysiological system using only efficacy data to enable a successful IND application into the FDA. Led to the authorization of a phase 2 clinical trial for rare disease. Began recruiting patients in April of 2021. And so this has now been a successful phase 2. It's now in a 2-arm phase 3 trial, which started in May of 2024. So slides are a little slow sometimes. And so how does these systems work? This whole idea of clinically relevant functional readouts I've been working on for about 30 years. And what does that mean? You go to a doctor's office, he doesn't immediately stick a needle in your arm to be able to pull out fluids, look at biomarkers, right? Looks how you're walking, how you're talking, you're making sense. He listens to your heart, he checks your reflexes. He might ask you about elimination functions. What he's really checking on is, are your muscles contracting okay? Is your electrical activity for neurons and cardiac cells functioning properly? Is your neuromuscular junction working well? Or basically the barrier integrity for your GI tract and kidney, is that functioning well? See, these clinically relevant systems are based upon biomems devices we integrated with cells, where you can put cardiac cells on top of cantilevers. So when they contract, they bend, deflects a laser, we can get force from that. We can pattern cardiomyocytes over microelectrodes to be able to get conduction velocity, QT interval. We can again do the same thing with skeletal muscle. A wide variety of neuronal systems I'll talk about. We can still look at biomarkers, for say liver, look at albumin production, CYP activity, but we don't have to. This allows us for non-invasive monitoring for acute, but more importantly, chronic activity in these human-to-chip systems, all without cell death. We're really trying to relate to what you see clinically, because if cells are dying, then a patient is in really bad shape. And how it works, what we can do is we culture out the cells on their own chips in their own conditions, and then we actually put them into a platform. We then put a gasket on top of it with the defined channels in it. And everything we use is all in serum-free media. I banished the use of serum in my lab 25 years ago. And so we put then a clear top on it so we can access all the different imaging tools in the systems. The nice thing is it's reconfigurable, so I can put different fluidic channels, the number of chips I have in there. I can also take out, say, this chip and put it pancreatic systems. I can also put organoids in the models and be able to evaluate them using imaging, say a cardiac organoid, and be able to look at the cardiac electrical data and force data at the same time monitoring the organoids to be able to then compare data somebody actually may have with the organoids as well. The other thing too is that Mike Schuler had taken physiologically based pharmacokinetic models, which are basically the in silico programs used to predict phase 1 clinical dosing, modified those, and then designed the first multi-organ system that was published in the, I think, early 2000s. This is a much more advanced system than we have now, looking at opioid overdose and recovery, at the same time looking at off-target toxicity. But they basically, all these systems use this idea of a pumpless platform. This basically, Mike patented the idea of using gravity to pump these systems. You put in a rocker board. What that does is it gets rid of all the tubing, all the pumps, makes it a simple system, gets rid of bubbles. But very importantly, it makes it also a low volume system. So if a liver present, I can look at the effect of both parent and metabolite in the same system. And this made the cover lab on the chip in 2010. And so it just is a very busy slide, but just shows how we can use lasers to define a pattern on top of microelectrodes, to pattern cardiomyocytes over top of that. You can see here a U-shaped pattern of the system. I can look at rhythm generation, conduction velocity, AP length or QT interval in the system. And on a separate chip, I can actually monitor force. Combined together, I can get cardiac output estimation. And this is published in Biomaterials in 2015. Allows us to look at spontaneous activity, okay, in the system, but more importantly, stimulate activity. So I can simulate here, be able to monitor activity over each of these electrodes, and then long stretches of the pattern. I can then pick it back up, be able to get conduction velocity in this system. In this particular experiment, it was 0.42 meters per second. In a human, it's about 0.5. But also I can look at QT interval at every point along the way. So I can then monitor that as well. So we've done well over 50 drugs with the cardiac systems. And this basically shows that we can take frequency, take norepinephrine, dose it with it. We can do a dose response curve. It speeds up the heart. Verapamil, which is a calcium channel blocker, which basically decreases the amplitude. Or look at something like Sodalol, where we don't see any effect on force, but we do see a QT prolongation. So I could basically get some mechanistic information of that because I would know all the pathways involved in force aren't involved in whatever toxicity this drug has. It was also electrical, and I can then do some further refinement of the model to be able to start doing some target identification. We can also culture out hepatocytes in this system. Now, these are just hepatocytes. These are primaries. We can culture them in 2D out for 28 days or 3D systems. Now, we can selectively add in stellate cells or Kupffer cells. We don't have to have them, though, to get the model to work. Yeah, we can also have nice, good basal activity for 1A1 and also 3A4, stimulate activity with these models. And again, out to 28 days. This is published in Biotechnology Progress. Now, AstraZeneca had a problem with a drug where it flew through preclinical trials. There's no safety concerns. As soon as they got into an animal, it had a huge QT interval effect. And so what we did is they said, well, let's validate the program, the platform first. Let's take terfenadine. Fexofenadine. Terfenadine is a antihistamine. It was pulled off the market many years ago because it had a QT interval prolongation, whereas its metabolite, fexofenadine, marketed as Allegrate now, does not. So if you see a cardiac-only system, you see a huge QT interval effect. We add in the liver, you see it shift to the right. Conduction velocity, basically is not affected if the liver is present. You see an IC50 of 33 micromolar, okay, if it's cardiac only. And again, we see this in terms of there's not any cell death in this model. Now, the next thing we can do with these systems, and we can then model them. Okay, we can grid up the system to be able to look at flow rates, shear stress, to be able to decide what is the right parameters for this model. We can then look at mixing in the system. We can also account for loss due to metabolism or for absorption in the systems. People don't realize plastics absorb molecules, and there's just no plastic that is universally does not absorb molecules. Some are a little bit better than others, but what we do to account for this is we simply just do control experiments to measure the amount of absorption, okay, and it goes into our model system for looking at the pharmacokinetics. So I then can account for the loss due to metabolism, due to absorption. I get the exposure area under the curve, and be able to create a pharmacokinetic pharmacodynamic relationship where I can account for the absorption, get the pharmacokinetics of that, account for metabolism, get the pharmacokinetics of that, combine them with the clinically relevant functional readouts to be able to get the pharmacodynamics of the cardiac model. Now, we can get that from the outside interaction of the hERG cell to really get the correct pharmacodynamics. Terfenadine actually binds at the hERG channel from the inside of the cell. This is the proper pharmacodynamics we're able to determine from this. And then be able to use that data to then combine it to predict guinea pig, dog, and non-human primate data with AstraZeneca, and then be able very good correlation with that. This was published in Nature Scientific Reports with AstraZeneca, basically showing that not only do we have good in vitro predictive capabilities, but we can extrapolate that to in vivo pharmacodynamics as well. Now, Roche challenged us with this model and said, well, okay, can you take that heart-liver system and actually add in an efficacy target at the same time? And they said, well, let's make it a little bit difficult. Let's make it a multi-drug resistant cancer and a non-multi-drug resistant cancer. You see, they picked tamoxifen. Tamoxifen by itself is not a very good chemotherapeutic. However, its metabolite is. So when you add in liver, we do see a decrease in proliferation. When you just look at its metabolite, you see a decrease in proliferation. In the multi-drug resistant cancer, you can see here no effect. Tamoxifen with liver, with the metabolite. But when you add in a second drug, verapamil, which blocks the PGP channel, which is pumping out the chemotherapeutic as fast as it goes in, you now see a decrease in proliferation. No additional decrease in proliferation in the non-multi-drug resistant cancer, showing verapamil was just acting as a PGP inhibitor in this model. Now, monitoring the off-target toxicity at the same time, for tamoxifen, you do see conduction velocity go down, but recover. Force go down and mostly recover. Beat frequency go down a little bit and recover. But for the drug-drug combination, you see that the conduction velocity goes down but does not recover in 3 days. You see force that goes down but barely recovers anything in 3 days. And beat frequency goes down, has some recovery. Now, importantly, if you're looking at an organoid-based system looking at beat frequency, you'll basically say, well, this combination is relatively safe. But you really have to have the functional readouts. And again, this is all done without cell death, which will basically say that this drug-drug combination is not feasible for therapeutic application. And that becomes really important, especially for cancer, where people have a pretty good idea how efficacy combines with chemotherapeutics, very little idea how the off-target toxicity combines in many of these systems. So now that cancer is going to lots of drug-drug combinations, it becomes really important to be able to monitor this before you actually go and scale this up to be able to go into tox studies in large animals. It gets hellishly expensive. We can actually screen this before that point. Again, you look in the liver system without any toxicity in this model. This is published in Science Translational Medicine with Roche and was featured in the NIH director's blog. Now, we've taken an extension of this model where we did a 4-organ system where we wanted to look at one of the compounds for Vanda Pharmaceuticals, tradipitant. They had been asked to do a 9-month Beagle study. They countered and said they wanted to do a 3-month Beagle study. They did a 4-organ model where we had control drugs for each one of the organ systems. And we basically combined that data with their 3-month Beagle study, and then it was allowed to proceed to review for the IND without having to do the 9-month Beagle study. So we're making inroads in terms of safety with these models. And there's other projects we have ongoing as well. So we've also been able to take the multi-organ system, create an innate immune system on a chip, where we basically have flowing monocytes in the model. And there should be a video here. But basically shows the monocytes flowing back and forth in the system without being activated by the flow or the presence of the other organs. Then when we basically selectively damage the cardiac cells, we're able to induce the M2 phenotype. And when we activated with LPS, the immune cells that got the inflammatory phenotype, we saw all the cytokine markers for the M1 phenotype. In summary, we can actually take this pumpless system, recirculating serum-free media, unique to Hesperos as well as the clinically relevant functional readouts. The systems we use for both efficacy and toxicity, and also at the same time. Reconfigurable and flexible platforms. We can create PK/PD models of these systems to be able to extrapolate, predict in vivo outcomes, and we can look at parent and metabolite in the same system without prior identification of the metabolite. Now, another model system we have is neuromuscular junction, where we can take human motor neurons on one side of a barrier and then human muscle on the other side, and have tunnels through the model. You can see here, and then be able to get innervation of the myotubes by the stem cell-derived motor neurons. And again, you see here, We can put the myotubes on cantilevers, okay, or we can look at motion capture here. We can stimulate the motor neurons, and then the signal goes through the axons, through the tunnels, into the neuromuscular junctions on the myotubes. We were first to be able to publish a dose response curve for Botox. Here, this was published in 2018. And again, showing that we can actually look at tox with this model. We are now been funded by NIH to be able to develop this as a FDA-approved model for Botox, because a lot of animals are sacrificed for this, and we're trying to eliminate that need. We can also use these systems for efficacy for the NMJ model. One of the first systems we were looking at for ALS, we've been able to differentiate out multiple SOD1 mutations for motor neurons and muscle and Schwann cells, as well as TDP43, FUS, and C9. And the idea, we could put the diseased motor neurons on this side and have the wild type muscle on this side. And again, in the wild type, you see good fidelity with in the system, less fidelity in the ALS models, but we can restore that. And so, again, getting back to this clinically relevant functional readout. So we had asked somebody who was running clinical trials in Mass General, well, what are some of the tests you have a patient do during a clinical trial to assess their function? Well, the one thing they have them do is a task with their hands faster and faster. And what they see is that they start getting jitters, okay, and they also start losing grip strength. Well, we know we can model that because I can stimulate the motor neurons faster and faster, in this case, 0.33 hertz, 0.5 hertz, 1 hertz, and then 2 hertz. And if I see any skipping of these signals, it's akin to the jitters they see in a clinical trial. Now, if I see loss of tetanus, that's akin to loss of grip strength in a clinical trial. It's a very, very busy slide, but this is one of our first publications in this area. Basically shows on the left-hand side, at the very top, you see the wild type has great fidelity, whereas on the two SOD1 mutations, you basically see some skipping and partial loss of tetanus with one SOD mutation, less skipping but complete loss of tetanus at the other SOD1 mutation. And for FUS, you see less skipping, but you see complete loss of tetanus. On the right-hand side, because it's wild-type muscle, when we stimulate on the muscle-only side, we see great fidelity as well as tetanus in these model systems. You look at the bottom, you can actually clearly see the statistical significance of the changes. Now, another equation directly out of the clinical trial is fatigue index, where we're basically applying tetanus to be able to look and see whether tetanus is still apparent. You can clearly see here that the fatigue index goes up, okay, with the mutations, as you see in a clinical trial. Now, we can also differentiate out Schwann cells using these models. This is with a SOD1 mutation. Basically, so we get all the correct markers. We get the Schwann cells, okay, with the mutation. And then a company, Apellis, asked us to create a inflammation model because their drug, pegcetacoplan, basically inhibits the complement cascade. So we actually took our bipartite system and created a quintipartite system by adding microglia on one side, and then Schwann cells in either unactivated monocytes or activated monocytes on the muscle side. You see, in the unactivated monocytes, you added in the complement sera, you see some reduction and an increase in number of neuromuscular junctions with the unactivated THPs. But with the activated monocytes, you see a huge reduction, but then recovery, okay, in this inflammation model. Again, for fatigue index, not much change in terms of their drug with the unactivated monocytes, but you see a huge reduction, okay, in terms of the fatigue index, okay, when you take their highest drug concentration. And you see a very good neurofilament light was used by Biogen to get an SOD1 treatment approved by the FDA. And then we basically show that in a molecule, we get an increase in neurofilament light with the complement, but then as you add in their molecule, you get a slow decrease in that model system. And again, this was used for successful IND application and orphan drug designation, and we're working on a paper on this data. Now, another model we've been able to look at this is myasthenia gravis. For those of you unfamiliar with myasthenia gravis, what your body does is it raises antibodies to the cholinergic receptors on the muscle. So basically, it blocks the neurotransmitters that are released through the synapse, or it causes internalization of the receptors, or causes initiation of the complement cascade. And we basically looked at the antibody to the cholinergic receptors, get a nice dose response curve. Other antibodies had no effect. We saw with the muscle, there was a nice window where we could actually then look at therapeutics, but at the highest concentrations, even the muscle started to lose activity. And we wanted to do is first off, look at the presence of the receptor on the outside of the cell. So we looked at it without Triton. We actually see that present. Basically, won't see anything with the higher concentrations, but actually with the with Triton, which basically permeabilizes the cell, we see them present on the inside of the cell, but at the highest concentrations, very little present. You guys can look at the quantification of that. You see them on both sides without the antibody. With the lower amount of the antibody, you see less on the outside, and then but some on the inside. You see that being downregulated for the highest concentration. This was published in Frontiers in Cell Development Biology. You've been also asked to take a look at Charcot-Marie Tooth disease. There are two main mutations, CMT1 and CMT2. But then we were asked to be able to do a clinical trial of one, the ultimate in personalized medicine, for a rare variation of that rare disease, CMT2S. So we took the fibroblasts from a patient, turned them into stem cells, differentiate out the motor neurons. We basically show that we got mature motor neurons with the model. We got nice chat staining and MAP2 staining. So we got mature motor neurons. Okay, and then for CMT, in the clinical manifestation does really not show those jitters you get with ALS, but it does show a loss of grip strength. You can see here we get the fatigue index going up. We don't see much changes in fidelity, but the fatigue index is significant at every treatment time. And what we do see is not only see this is normal tetanus, we see a change in tetanus, which is kind of abnormal. And if we then, what they wanted to do was create an antisense oligonucleotide for that particular patient's mutation. And so when I say chaotic tetanus, what we did is we seen, going to tetanus and then going out and back in and stuff. And then having it, these are all single myotubes. Now in the muscle itself, you have a bundle of myotubes. So if you sort of sum all these up, you probably get 40 or 60% of function, which is what a CMT patient sees. But they also have sudden falls. And if you think about, how these myotubes kind of sum up, there might be a time when all of them kind of fail at the same time, which would be loss of tetanus, and then maybe call this a falls. We think we're pretty good predicting the clinical manifestation of this. And then we treated the patient with the ASO. You see pretty much normal tetanus restored in just about all the myotubes. And this is actually entering into the clinical trial now. And we can also have looking at myelination. We've been able to create myelination models with this system. This is published in ACS Chemical Neuroscience with primary Schwann cells. We now have a paper that is accepted with induced pluripotent stem cell derived Schwann cells with the induced pluripotent stem cell derived motor neurons. But a small company in California had seen these results and said, could you do a conduction velocity model for two rare diseases, multifocal motor neuropathy and CIDP, where what your body does is it raises antibodies to nodes around VA, basically shuts down your conduction velocity because it activates the complement cascade, okay, which basically forms pores at the nodes around VA, which basically then stops your clinical conduction. And what they did is they had a molecule that interfered with the C1 complex. So what we did is we took patient sera samples. So we could take mutations to create a disease model, but also patient sera samples. Basically showing here complement deposition with two patient sera samples, but their molecule then abolished that. We saw positive results where we got the not much C3 or C5 present for 15 different patient samples. But again, we want to stick with this idea of clinically relevant functional readouts. This is very straightforward. It's conduction velocity. That's what's measured in the patient. So we basically took the neuromuscular junction model, turned it 90 degrees. Now we put the electrodes inside the tunnels so we can measure conduction velocity. So what we want to try and do is be able to measure over 3 electrodes. So we can actually then tease out the effect in individual axons. So for 2 electrodes, we get a large number, but we still get a significant number of conductions over 3 electrodes. And again, you get a range of conductions. This was the key experiment. What we did is we did a pre-compound test. We did the untreated. We did the serum plus the isotope control, but the serum plus their molecule. Then we saw a range of conduction velocities in the control after the compound. We saw conduction abolished with the serum plus the isotope control, but with their molecule, we saw that was protected. And so this was the data, went into the IND, enabled this phase 2 clinical trial, which is now proceeded into a phase 3 clinical trial. So not only getting regulatory acceptance of these models, but also we're getting, it looks like clinical predictivity, at least in a small sample set. This was published in Advanced Therapeutics with Sanofi, which acquired that small company. Also, I forgot to mention that for the myasthenia gravis model, that model was used by Dianthus to be able to get their therapeutic into a clinical trial as well. Summary of main points. We developed models of peripheral nervous system that successfully recapitulate clinical readouts, can be used for both toxicity and efficacy. NMJ models can be used for successful IND applications as well as for conduction velocity. We demonstrated we can actually do a personalized medicine with these models. And the system could reproduce clinical functions so well that mechanistic insights can be gained that were not previously realized, as we've seen with the CMT model. And lastly, I want to go through some work that we have with a CNS model. This is a cartoon I put together about 20 years ago now, where my idea of looking at long-term potentiation was to take hippocampal neurons and integrate them with microelectrodes, and then be able to look at the act of storage and retrieval out of cortical neurons. And so what I'm really trying to do is to reproduce the dual patch clamp and slice, which is the basis for a lot of research in long-term potentiation. And so what they really could do with that is they put a micropipette in one part of the slice. They stimulate. They have another micropipette on a neuron in another part of the slice, and they record. So what they can do is they can actually separate the neurons from each other, but they still have axonal connections where they can simulate one and see what happens in the other. So I can do that with MEAs if I can use surface patterning, right? So I put down modulators. I can put down silanes, put a mask over it, shine light on it, remove the silane, and come back with a second silane to make patterns. This was first published in 1998. Okay, so this is a more advanced pattern. What I'm trying to do is I'm trying to get the neuronal cells to go to the somal adhesion points, then put out neurites to become the axons and dendrites, and I have a control where they're coming from. A movie should be playing. There we go. It's just slow as there's a video. Or it's not, you're not going to be able to see it at all. I'm not really sure. All right. So what you should see is 2 neurons putting out the pathfinding processes, finding the somal adhesion sites, and then self-assembling onto the pattern where we control which one become axons and which ones become dendrites. And so this is actually an example of the patterning where we take now a population of neurons, which is not possible with dual patch clamp and slices. And then we can put them over one population over one set of electrodes, another population of another set, use surface chemistry to connect them through the axons. So I can stimulate here, record here, which is basically structure for you have for dual patch clamp electrophysiology. Now, very important for MEAs it's real easy to make MEA noise look like a neuronal signals. So what we did is we actually hit, after every experiment, hit it with lidocaine, basically, so we can actually figure out what is noise and what's biological. So we can, and this was published in Stem Cell Reports. And the other thing we needed to do was show we actually had mature human cortical neurons. So we developed the differentiation protocol, and we basically showed that the even though we had proper staining for the cells, we weren't able to induce LTP because we could stimulate but wasn't maintained. After 35 days, we could stimulate, it was maintained. So at this point, we have maturation. We have also the right ICC for this. But also as a demonstration of control of the systems, one of the things we realized that the immature cultures had a population of inhibitory as well as excitatory. So when we actually block the inhibitory signals, we're able to then induce LTP in these models as well. And again, we wanted to prove we had LTP. AMPA receptors are intimately involved in LTP, so we could see that before we blocked them, we were able to induce LTP. After we blocked them, we were able to knock that out. Same thing with NMDA receptors. We use a different inhibitor, DAP5. Again, before application, we can induce LTP, and this was actually reduced, actually eliminated after the molecule to block the NMDA receptors. So we have good data to support the fact we can do LTP in these models. We're using this for Alzheimer's disease, where we can take A-beta scrambled. A-beta oligomers or soluble oligomers are thought to be the toxic species in A-beta. So again, we can then treat it and induce LTP. The oligomers then abolish that. We can do the same thing with tau fibrils. Again, the control maintain LTP. We abolished it. This is published in Alzheimer's and Dementia. And again, this is all done without cell death in these systems. And then again, to show the 1-1 correlation, we able to do the patch clamp with the A-beta scrambled. See nice currents, good induced activity, good spontaneous activity. A-beta 42 oligomers pretty much abolished the currents mostly, the induced activity, and any spontaneous activity. But when we took an AD drug, approved drug, donepezil, we were able to show that we restored currents to some degree, or induced current, and also spontaneously firing. But the key thing is you see a 1-to-1 correspondence with the data on the patch clamp on the MEA systems. We're able then to do reproduce this with 4 different drugs. The 2 that were basically approved by the FDA for Alzheimer's, 2 that are being used for off-label. Where again, A-beta oligomers were abolished LTP. It was being restored with the drugs in each case. And this paper has just been accepted into scientific reports. And lastly, showing how the CNS model can also be used for toxicity. Are those familiar with the idea that a lot of drugs have black box warning labels saying don't operate machinery, don't drive a car? That's because a lot of them have activity that reduces your cognitive function. One of the class of molecules that are famous for this is anticholinergics, Benadryl being the one that most people are familiar with, which is a sleep aid. But there's actually a scale developed for anticholinergics that pharmacists and clinicians use to be able to say, well, okay, should this drug be prescribed on certain disease conditions or with other certain drugs? And you see here that this is a 0 to 3 scale based upon anecdotal evidence from clinicians. You see at 0, we didn't see much effect. We get slight dose response curves for ACV1. ACV2, we get more of an effect. ACV3, we see a big dose response curve for these molecules. And look at that, you see a family of curves for 1-hour LTP and then also 24-hour LTP in these model systems. And this is actually its correlation with the ACV scores, showing pretty good correlation, although there are some molecules are kind of like a little bit out of sync. And clinicians have been complaining about the scale for a long time, say it's not particularly accurate. And this kind of just shows that there are some variations. We're working now with a company to integrate these predictive models with their in silico models to get much more predictivity for at least the anticholinergics and extend it to other drug classes as well. And the last thing is basically when you wake up after taking Benadryl in the morning, you wake up, you're feeling groggy. The first thing you do is reach for caffeine. I said, well, can we actually really reproduce a clinically relevant readout with this? So we actually look at caffeine. Caffeine has some small effect. But the anticholinergics you see, it's actually wiping out the LTP, but the caffeine restoring it. For diphenylhydramine, which is Benadryl, you see it knocking it out, but then restored. Okay. Now, scopolamine, which is a much more powerful anticholinergic, you see basically it doesn't restore it until we get a really high concentrations of the drug. So summary main points: we have a CNS model of LTP, correlates with the cognitive function, using investigative work to lead to IND applications. Can utilize both efficacy and toxicity. Use a variety of different applications of disease models, toxicity determination, and cognitive function. I didn't go into it. We also have a stress model for the system. But the main thing with the CNS model, it can also be integrated with the multi-organ platforms I showed in the beginning of the presentation as well. So we can actually integrate that. We've shown with GI tract and BBB and various combinations thereof. So basically, I hope we have given you a pretty good idea of the work that we do at Hesperos, but also in my lab. We can create functional multi-organ platforms, determine therapeutic index with those systems, reduce the need for animal testing. And really what we can do is our business model is we can custom build whatever the customer wants in terms of these therapeutic models. Thank you for your virtual attention. And again, the ways to contact Hesperos if you're interested in more information are listed at the bottom of the slide. Thank you very much.